different microbes. Okay, this chapter give you idea about what is the use of the microbes, what are the use of bacteria, fungus and other microbes in our daily life. Okay, just like your lactobacillus acidophilus clostridium, okay, botulicum, that is your acetobacter aceti, different bacteria, fungus, viruses, microorganisms that can be used in our life. Okay, daily life practices in factories, in making antibiotics, in sewage treatment plant, in biogas production, different areas in this world, different sectors in the world, we use microbes in daily basis. Okay, basically microbes causes disease. Okay, but just like that every coin have two faces, one is head and one is tail. हर सिक्के के दो पहलू होते हैं, एक होता है हेड और एक होता है टेल. ओके, ऐसे ही माइक्रोब्स के भी दो पहलू हैं एक इंसान की जिंदगी में. ओके, देयर इज टू फेसेस ऑफ माइक्रोब्स इनसाइड द लाइफ ऑफ ह्यूमन. ओके, आइदर इट कैन बी used for a better purpose or it can cause disease. Okay, just like your tuberculosis bacterium, mycobacterium tuberculosis, mycobacterium leprae, all these bacteria causes diseases. But on other hand, some bacteria, some viruses, some fungus are there which is used for the welfare of human. So in this chapter we are going to discuss all these microbes which are going to be used in our daily life and which is used for the human welfare. Okay, so let's get started. First, this chapter is divided into 5 parts. First is related to microbes in household. Okay, second one is microbes in that factories, in industry scale. Second, microbes in your that biocontrol. Okay, in farmer fields. Okay, fourth one, microbes in biogas production, and fifth one in microbes used in sewage treatment. Okay, so first we have microbes in daily life. Okay, so microbes are the tiny living organism that cannot seen with your naked eye. Okay, you cannot see them by your naked eye, you have to apply microscope to see them. Okay, these are not visible by your naked eyes, you have to use microscope to see these microbes. Okay, so these microbes are present not only in the human body but all around the life, like soil, in water and air. Microbes are present everywhere, in soil, in air, in water. Even some microbes you will find in hot water springs, volcanic areas, deep sea, highly saline waters. These are your archaebacteria. Okay, so all these bacteria, microbes, fungus, viruses, you will find them everywhere in the world. Okay, some are found in most extreme habitat like soil buried below many layers of snow. Some are also found near the deep sea hydrothermal vents with temperature reaching up to the 100 degrees Celsius. One can observe them with the help of microscope, then they are named as microorganism. Why we call them microorganism? Because we cannot see them by our naked eyes. We have to see them by microscope. That's why we call them microorganisms or microbes, as they need to be magnified over manifold times to become visible. Okay, when you have to see these organisms, you have to apply microscope, and by the microscope you can see these organisms. Without microscope you cannot see these organisms. That's why we call them microorganisms. Okay, so these are the microbes which you can see by different axes on microscope. Just like bacteria, you have to magnify your microscope up to 1000x. For fungus, again 1000x. Okay, then your protozoa 1000x. Pyrones, you have to magnify your microscope up to 1 lakh x. And for that viruses is also 1 lakh x. Okay, why these pyrones and viruses require that much magnification? Because they are smaller than your bacteria, fungi, protozoans. Okay, so microbes magnified by 10, 1 lakh or 15, 1 lakh 50,000 x. Example of virus include adenovirus, tobacco mosaic virus and bacteriophage. This is your adenovirus, this is your TMV, this is your bacteriophage. These all are different categories of virus which you studied in biological classification in 11th class in the virus part of that chapter. Okay, so now where you will grow these microbes? What is the area where you can grow these microbes? That area is petri dish or culturing medium. Okay, so microbes are too small to see directly. Bacteria, maybe fungi can grow on nutritive medium. That is your culturing medium. Okay, comparison of nutritive medium, water, agar-agar, that is solidifying agent which is used to create from red algae, gelidium and gallisneria. Okay, then salt, sugar, vitamin, amino acid, these all are nutrients which you give to that organism, microorganism, to grow in that petri dish. Okay, you can see here, this is your nutritive medium or your colonizing medium, okay, in which you have these things: water, agar, salt, sugar, vitamin, amino acid. All these things come together and give you that proper house, proper medium in which your microbes can easily grow. Okay, so petri dish with nutritive medium, in this they will easily grow like this. In this you will see this white blasting type area. This is the colony of fungus penicillium. Okay, so the microbes multiply rapidly on nutritive medium and form dense group of cells called colonies. Okay, these are not a single, these are the number of that microorganism living in a single area. That thing is called colonies. Okay, colony of bacteria, colony of fungi. Okay, so these are the group of organisms which live in that particular area. So these colonies are visible to naked eye. Okay, now these are in millions in number in that colony, so you can see them by your naked eye. You do not require microscope to see them because they are present in large number in that particular area. Okay, so fungi can also grow on nutritive media. Cells of fungi multiply rapidly and form dense group of mycelium called colonies. Okay, that mycelium. Okay, mycelium, what is mycelium? Mycelium, when that hyphae group together and they form a structure which is called mycelium, and mycelium you will see in fungus. Okay, upon microscopic observation the hyphae and spores of fungi can be seen, but if you want to see the hyphae and spores of fungus, then you will use that microscope. If you see the mycelium of fungus, then you can see that by naked eyes, but if you want to see the spores and hyphae of that fungus which is growing inside that medium, nutritive medium, then you have to use microscope. Okay, so bacterial colonies are also growing, appear small and rounded. Bacteria can be in many shapes, but the most common ones are spherical shape, which is called cocci, and rod shaped bacilli. In biological classification chapter, you studied that bacteria is divided into 4 groups on the basis of their shapes. That is your coccus, which is round shape, then bacillus, rod shape, comma shape, vibrio, and spiral shape, spirulinum. Okay, so in basically that spherical, that is your cocci, and the rod shape bacilli, that is the most widely used in the welfare of humans. Okay, the microbes which is used to produce different substances for human, they are mostly spherical shape and rod shape. Okay, so first use of microbe which we are going to study in today class is curd, dahi. Okay, that thing you drink every day. Okay, you use every day, you drink every day, that is your curd. Okay, so curd has lactobacillus bacteria and it is also a fermented product. Okay, see, lactobacillus, okay, it is an bacillus bacteria, okay, which also called as lab, lactobacillus acidophilus, lab. Okay, so lactic acid bacteria lab include lactobacillus lactis, lactococcus lactis, and lactobacillus vulgaris. Okay, so these are the lactic acid bacteria. These bacteria produces lactic acid in the presence of milk, means they convert milk into curd by secreting out lactic acid. Okay, and these are the 3 bacteria which do this thing, that lactobacillus lactis, lactococcus lactis, and lactobacillus vulgaris. Okay, the lab are unique, they thrive the acidic environment which allow them to outcompete and any other bacteria in its environment. Okay, then see, now as you studied ecology, competition is everywhere. Okay, so competition is in that milk medium is also when that lab is present there. Okay, so how they will compete out that competition? They secrete out acids, lactic acid. That lactic acid does not allow other bacteria to grow in that medium, and that rapidly multiply the number of lactic acid bacteria, your lactobacillus acidophilus bacteria. Okay, so the food fermented by lab is also productive from harmful bacteria. Okay, now automatically these lab bacteria secreting that lactic acid. Okay, now other organism does not come, so that acid protecting these bacteria and either also protecting its area from other bacteria. Okay, so the food fermented by lab is also protected from harmful bacteria. So, okay, in anaerobic condition they are perform lactic acid fermentation for energy production. Okay, in 11th class there is one chapter, respiration in higher plants. There you study two types of respiration, aerobic respiration and anaerobic respiration. Aerobic respiration means when you require oxygen to do respiration, and anaerobic respiration means when oxygen is not required for doing respiration, oxygen is absent in that process. So these bacteria do anaerobic respiration, they perform their work in anaerobic conditions. Okay, so in anaerobic condition they perform lactic acid fermentation for energy production. Okay, and that in that chapter you studied that glycolysis happens. The end product of glycolysis is pyruvate. Now pyruvate have two fates. Okay, if it will go in aerobic respiration, then it will enter into Krebs cycle and ETS. Okay, and if it goes in anaerobic respiration, then it will having two fates. Either it will start to produce alcohol or it will produce lactic acid. So these bacteria use that lactic acid to make energy. Okay, lactic acid is the source of energy for these three species of bacteria: Lactobacillus lactis, Lactococcus lactis, and Lactobacillus bulgaris. Okay, so what is the process of curding the milk? The process in which you will convert milk into curd is called curding of milk. Okay, the process of curd formation. So first you have to take warm milk in porous, poured into a clean container. ओके तुमको दही बनाना सिखा रहा हूं बेसिकली मैं, तुम घर पे दही कैसे बना सकते हो, हाउ कैन यू प्रिपेयर कर्ड एट योर हाउस. It is very easy. Warm milk is poured into a clean container. Okay, now warm is required for the fermentation process. Okay, warm condition is very important to perform fermentation process. Okay, milk is a mixture or emulsion. Okay, in chemistry you studied that different type of solutions: emulsion, sol, aerosol, solid, semi solid. Okay, so your milk is a emulsion. Okay, so in milk you will find fat globules, that is triacetylglycerol plus fatty acid plus cholesterol plus trace amount of fat soluble vitamins. Okay, that thing which is present inside the milk in the form of fat globules, it is your triacylglycerol, free fatty acids, then cholesterol and trace amount of fat soluble vitamins. Okay, second, micelles of casein protein, the protein which is present inside the milk is casein. Okay, so the milk micelles of casein protein, most abundant protein in cow milk, 80%. Your cow milk contain 80% casein protein. Casein protein is the sole member and the most important protein in milk. Okay, so soluble whey protein, that is 20%. When you do drying of milk, the powder form of milk is called whey and it is protein. Okay, whey protein. So protein 20%, calcium, phosphate, lactose and other molecules are also present inside your milk. Okay, to this milk a spoonful of curd is added called inoculum. Okay, so now in milk. Okay, so in milk now how will you make curd? You will add inoculum. Okay, inoculum is a small amount of curd which is added inside to milk, that to convert that milk into curd. Okay, so to this milk a spoonful of curd is added called inoculum, which is also called as starter, which start the process of curding of milk. That's why it is called starter. Two names are there, starter and inoculum. In Hindi we call this jam. Okay, so you add this inoculum inside that warm milk and now your warm milk start to convert into curd after it gets favorable condition to grow. Okay, and the process is called inoculation. Okay, the process in which you are adding inoculum inside the warm milk to convert that warm milk into curd is called inoculation. Okay, when curd contain bacteria is added to the milk, these bacteria get lot of milk sugar to feed on. The milk sugar, the lactose which is present inside the milk, that bacteria lactobacillus coccus, okay, bacillococcus, that coccus bacterius lactalis, okay, lactobacillus acidophilus, these bacteria start to consume these glucose, that sugar which is present inside the milk, that is your lactose, and then digest that thing, okay, and start to convert your milk into curd. Okay, so lactose sugar is taken up by a leg. Okay, as you know, the abundant sugar which is present inside the curd is lactose. lactose. Okay, so lactose sugar is taken up by the lab use lactose and convert into glucose and galactose. Okay, means when your lactose break down, it will convert into glucose and galactose. Glucose and galactose undergoes glycolysis to give pyruvate. Okay, you studied that thing in respiration. Glucose टूटेगा, दैट लैक्टोज टूट, उसमें अपन ने पढ़ा था कि सुक्रोज है अपने पास, सुक्रोज टूटेगा ग्लूकोज और फ्रक्टोज में. ओके, इन दैट रेस्पिरेशन in in angiosperms respiration in plants we studied that your sucrose will break down into glucose and your fructose. Now your lactose in milk break down into glucose and galactose. Okay, now these breakdown product glucose and galactose will go under glycolysis. Okay, and the end product of glycolysis, you know, that is pyruvate. Okay, now this pyruvate get anaerobic conditions, that condition in which oxygen is absent, anaerobic condition. So under anaerobic condition, pyruvate is converted into lactate 3 carbon compound which result in lactic acid fermentation. Basic concept, basic thing, you have milk, in milk you have a sugar which is lactose. So these bacteria do what? They digest lactose, they break down lactose into galactose and glucose. Okay, now this glucose and galactose will go under glycolysis and convert into pyruvate. Now this pyruvate will go under anaerobic respiration, anaerobic fermentation, in which it will convert into lactate, which is your 3 carbon compound, and after that lactate will convert into lactic acid, and the process is called lactic acid fermentation. Okay, see in the diagram you can see also lactic acid fermentation process. This is lactose breakdown into glucose and galactose, glycolysis take place, then pyruvate is produced, pyruvate go under anaerobic condition and will convert into lactic acid. Okay, so process of curd formation using lactose for anaerobic respiration, lactic acid fermentation, the bacteria divide rapidly. Okay, when the condition become that much anaerobic that bacteria start to replicate. Okay, this lead to increased production of lactic acid. Okay, this lactic acid decreases the pH of milk from 6.7 to below 4. Okay, in the starting your milk pH is 6.7 when there is no bacteria like lab. Okay, but when this bacteria, lab bacteria, lactobacillus acidophilus bacteria, they start their working inside the milk, then the pH of your milk from 6.7 it drop down to 4, which is an acidic pH. Okay, within 6 hours at room temperature. On your 37 degrees Celsius temperature, this pH of 6.7 of your milk drop down to 4 temperature within 6 hours. Okay, that is your how curding of milk take place. Okay, and second thing, this is how these bacteria do. When the pH is decreased, so what effect take place on the proteins which is present inside the milk. Okay, so this is your casein protein, it will unfold it. Okay, this is whey protein, it is get also unfolded. Okay, so decreasing pH causes the casein protein and whey protein to slowly denature, means these proteins start to denaturation, they goes to the step of denaturation, they are unfolding. Their decrement started when the pH of milk decreases. Okay, when your pH of milk goes to acidic, what thing happen? The protein which is present inside your milk, casein protein and whey protein, they will go down and they start to denature. Okay, so these denatured protein come together due to the hydrophobic interaction, electrostatic interaction and hydrogen bond to form a network resulting in coagulation and curdling. Okay, so the reason behind the curdling, the thickiness of the curd, okay, the thick texture of your curd is because of degradation, denaturation of the proteins which is present inside the curd, that casein protein and your whey protein. Okay, so the denatured proteins comes together due to hydrophobic interaction, electrostatic interaction and hydrogen bonding. Okay, and that's why your curd get that much thick curly texture. Okay, so no covalent bond like peptide or disulfide bond are broken. Okay, the disulfide and that your covalent bond which is peptide bond which is present inside the protein, they are not broken down. Okay, they are present inside that milk. Okay, that curd, that milk which is you converting into curd, it will contain peptide and disulfide bonds. Okay, So what are the benefits of drinking curd? Okay, you know that thing, but still my role, my work to tell you that what is the effectiveness to drinking curd in a day. So first, increase nutrition value of vitamin B12. Okay, our gut cannot absorb vitamin B12. We cannot produce vitamin B12 by our own. So where the source of vitamin B12? That is your curd. Okay. Second, easy to digest. Okay, it is already digested by lab. So how can you digest that? So it is easy to digest. Okay, already you are eating a digested food by a bacteria, then that's why it is easy to digest. Okay, third, probiotic gut health improves. See, in our gut, two types of bacteria are living: good bacteria and bad bacteria. When the good bacteria number increases inside your gut, you are healthy. When bad bacteria number increases inside your gut, you become unhealthy, you get diseased. So these bacteria, lab bacteria, they are good bacteria and they increases the healthiness of your gut. That's why it is probiotic. Okay, safe to consume for lactose intolerance people, because your lactose is already break down into glucose and galactose. So there is no lactose inside that thing. So the person who having lactose intolerance or lactose allergy, which cannot absorb lactose easily, which cannot digest lactose inside their gut, so they can easily absorb curd. Okay, because lactose is already break down into glucose and galactose, then you can easily absorb it. Okay, enhance texture and flavor. Okay, you can enhance the texture and flavor by adding different things inside the curd, but it does not affect the nature of the curd. Okay, so these are the benefits of eating curd daily. Okay, next is your idli and dosa. Okay, these are also fermented food which is produced in daily household life. Okay, so the holes and crispiness of dosa and fluffiness of idli are also due to the lactic acid fermentation. Okay, you see your mothers doing making lactic acid. First, what she will do. Okay, they put the rice, dals inside a container by straining the water and keep that thing overnight. In overnight, the fermentation of that rice and the dal take place. Okay, so rice and black gram are soaked into the water overnight and ground to make the batter. Okay, when you put that thing overnight, the bacteria which is present inside that water, the bacteria which is present inside that batter, it start to do fermentation. And after, when you do the grinding of that thing, you get a batter which is highly fermented. Okay, it contains bacteria, it is fermented, this excluding out CO2. Okay, so fermentation of dosa batter is carried out by mainly by lactic acid bacteria. Again, same bacteria which is present inside your curd, lactic acid bacillus, lactic acid bacillus. Okay, lower lab bacteria. Okay, so lactic acid bacteria present in the black gram release lactic acid and thereby leaven the batter. Okay, the lactic acid, where this lactic acid coming in curd, you add inoculum. Okay, in the curd you add inoculum inside the milk. That's why your milk convert into curd. But in this, now no inoculum you have to add. Okay, because that bacteria, lactobacillus bacillus bacteria, it is already present inside the gram. Okay, that black gram. Okay, and when you put that thing overnight for fermentation in anaerobic condition, it will start the fermentation of that batter. Okay, so that's how your crispy dosa and fluffy idli look like. Okay, next is your microbes in making cheese. Okay, cheese you already know. You eat that thing in your fast foods. You crush them, you grind them and put on the fast food. Okay, that cheese, mozzarella cheese, different type of cheese are present in the world. Okay, that French, France, Italy, they are famous for their that cheeses. Okay, so cheese is one of the oldest food item in which microbes are used. Okay, it is using from Egyptian time, from Greek. Okay, from Greek time, from Egyptian time, they are using cheese, and it is the oldest fermented food that is eaten by human. Okay, different varieties of cheese are known by their characteristic texture, flavor, and taste, and the specificity common from the microbes used. Okay, the size, the shape, the texture, the taste of the cheese, it is not from a single organism. It is produced by multiple microorganisms which is used to produce these cheeses. Okay, so we have in our course we have three cheeses: Swiss cheese, Roquefort cheese, and Camembert cheese. Okay, so first we have Swiss cheese. Okay, multiple bacteria involved in Swiss cheese production. The lab convert lactose to lactic acid. That is normal thing. Okay, then your main organism, Propionibacterium shermani, used to this lactic acid and produce CO2. Okay, Propionibacterium shermani, which is used to produce your Swiss cheese. It use was, it will do that lactic acid, okay, and produce CO2. It eat that lactic acid present inside that and it produce CO2. Now when this CO2 come out, it will make hole inside these cheeses. Okay, the hole which are you see in Swiss cheese, it is because of that CO2 which is producing by the fermentation of this bacterium. This bacterium do fermentation, and it, due to the result of that fermentation, CO2 gas evolved from the cheese and the cheese get these holes. Okay, so this is your Propionibacterium shermani, use lactic acid and produce CO2, propionic acid and acetic acid. These release CO2 bubbles are the reason of hole in this cheese. Okay, these holes, these are the cause. These are because produced when the CO2 gas evolved from the cheese. Okay, it is asked in NEET many times. See what gas is responsible for the holes inside the Swiss cheese, and the answer is CO2, carbon dioxide. Okay, so this is the basic concept. This is the basic flow chart for your how your that cheese is produced. So lactose, it will convert into lactic acid by your lactobacillus acidophilus bacteria. Now your Propionibacterium shermani will come in the play, and it convert this lactic acid into propionic acid and carbon dioxide, and this carbon dioxide is responsible for the holes of Swiss cheese. Okay, so this is your Propionibacterium shermani, and in the cartoon Tom and Jerry you see Swiss cheese every time. Okay, the cheese which is eaten by your Jerry is your Swiss cheese. Okay, the cheese, this your Tom Jerry is taking, this is your Swiss cheese, and this you will see the holes, and this holes is produced by CO2. Okay, the production of CO2 is responsible for this holes in Swiss cheese. Okay, now second one is your blue cheese or Roquefort cheese. Okay, Roquefort cheese का common name होता है blue cheese. Okay, so Roquefort cheese, a type of blue cheese, is made from sheep milk in France. Okay, it is made in France. It is a French cheese. Okay, you will find this the proper then the most tastiest cheese of Roquefort type in France. French. Okay, so Roquefort cheese, a type of blue cheese, is made from sheep milk in France, and it is made by sheep milk. Okay, it is made by using Penicillium roqueforti. Okay, so first we have Swiss cheese organism which is used to produce is your Propionibacterium shermani. Second cheese is your Roquefort cheese or blue cheese. It is produced by fungus Penicillium that roqueforti. Okay, Penicillium roqueforti is used to produce your Roquefort cheese or your blue cheese. Okay, so first the milk is pasteurized to kill any unwanted microbes. Okay, it is the basic step. First you have to pasteurize, first you have to clean the thing, then you have to add, then you have to use that thing. Okay, because you have to make the medium sterile, avoid the bacteria which is not is in your use. So first you do pasteurization of that milk. Okay, after this step the lactic acid bacteria are added along with enzyme called rennet. Okay, rennet is an enzyme which is fasten the process of curdling of milk. Okay, rennet hydrolyze casein protein allowing it to form gel like structure. Okay, rennet क्या करता है, casein के protein का hydrolysis कर देता है, and due to that hydrolysis your that milk become just like a jelly substance. Okay, so this solid mass of denatured protein are called curd. Okay, normal curding process we do not use rennet, but if we want to do that thing faster, then we add rennet enzyme inside the curd, and it fasten the process of curding, and the end product you get, it is your curd. Okay, now this curd is that in that curd the liquid part it is removed. Okay, the liquid part of curd is removed. The solid part, it is a semi-solid. Curd is a semi-solid. Okay, so the liquid part of that curd is drained out, removed. The solid part of curd is used. Okay, so the liquid is drained from the curd. Next, penicillium roquefort is sprinkled on the cheese. The cheese is allowed to age and ripen for 60 to 90 days. After 2 months or 3 months, you will put that cheese for ripening. Ripening means that fungus can grow and give its taste to that cheese. You put that cheese for 60 to 90 days means up to a 3 month. Okay, after 3 months you will find a thing which is your Roquefort cheese. That's why it is called blue cheese. You will find that color, that these are the colonies of fungus which is growing inside the cheese. That's why it is called blue cheese or Roquefort cheese. Okay, so the thing is, first you take milk, you pasteurize the milk. Okay, after that you add rennet inside that enzyme which hydrolyzes casein protein. Now that thing, the produce after that hydrolysis of casein protein is called curd. Now you remove the liquid from the curd and use the solid part. Okay, now you sprinkle the spores of penicillium roqueforti on that solid part and put that thing for aging for 30 to 90 days, means for 2 to 3 months, and after 2 to 3 months you will got this as a product which is your Roquefort cheese, and under microscope when you see, this is your penicillium roqueforti. Okay, so this is your second cheese. Now the third cheese is your Camembert cheese. Okay, so Camembert cheese is made from cow milk. Okay, Roquefort cheese is produced by sheep milk. Camembert cheese is produced by cow milk. Okay, it is made using mold of Penicillium camemberti. Swiss cheese we use Bacterium, Propionibacterium shermani. Roquefort cheese we use fungus, that is your Penicillium roqueforti. Now third cheese is your Camembert cheese. Again we are using a fungus, that is your Penicillium that camemberti. Okay, so it is known as for smooth and goosey texture. Okay, it is not hard cheese. Okay, चीज की दो कैटेगरी होती है, एक होती है हार्ड चीज जस्ट लाइक योर स्विस चीज एंड योर रॉकफोर्ट चीज, एंड अदर कैटेगरी इज सॉफ्ट चीज. बाय applying a simple pressure on that cheese, it will melt down, it will break down. Okay, so that type of cheese is your Camembert cheese. Okay, so it is known for smooth and goosey texture. Okay, the fungus releases hydrolytic enzyme and digests and soften the cheese from inside, thereby leading to this texture. How this texture coming inside that cheese? It is coming by the help of the enzyme out from your fungus. The hydrolytic enzyme that fungus is releasing, it giving it that texture, that soft texture. Okay, so this is your Camembert cheese, and this is the organism which is used to produce Camembert cheese is Penicillium camemberti. Okay, so till now we get three cheeses. Swiss cheese produced by a bacterium, Propionibacterium shermani. Second one is Roquefort cheese produced by a fungus, Penicillium roqueforti, and third one is your Camembert cheese produced by a fungus again, that is your Penicillium camemberti. Okay, so this is your cheese. Okay, next third product which is produced by the help of microbes, that is bread. Okay, bread, yes, bread that you eat in breakfast daily. It is also produced by the activity of a microorganism known as yeast. Okay, Saccharomyces cerevisiae, baker yeast. Okay, so the dough which is used to making bread is fermented by baker is called Saccharomyces cerevisiae. Okay, the scientific name of yeast is Saccharomyces cerevisiae. Okay, so to make bread, wheat flour plus sugar plus salt plus oil plus water and yeast are added. Okay, this is the recipe to make bread. Okay, wheat flour, atta, sugar, salt, oil, water. Okay, again I am saying the recipe to make bread: wheat flour, sugar, salt, oil, water is the most important ingredient for the fluffiness and the porousness of bread coming is from yeast are added. Okay, wheat flour has a protein called gluten. Okay, gluten protein which is present inside the wheat. Now your days are listening gluten free food, gluten free bakery in which you will not find wheat some other alternative of wheat, some other cereals which is can be used. Okay, so this is the protein which is present inside the wheat. It is gluten. Okay, so when water is added to the flour and knead, gluten forms meshwork. Meshwork means जब तुम नॉर्मल आटे में पानी डालते हो तो आटा गूंथता है, तो दैट slimy solid thing, okay, that slimy thing, it is your meshwork. Okay, so this meshwork gives this, you will get a chewiness. It is come because of that meshwork. Okay, yeast tangle in this mesh and utilize simple carbohydrate like sugar and convert them into ethanol and carbon dioxide by anaerobic respiration. In previous you studied lacto acid, that curd, it is using process lactate, that lactic acid it is producing, okay, by fermentation. In this you will see your yeast produce ethanol, so it is doing ethanolic fermentation, while your lab bacteria they do lactic acid fermentation, your yeast doing ethanol fermentation. Okay, so yeast is tangled in the mash, utilize the carbohydrate which is present inside the mash, and in anaerobic condition it converting that carbohydrate into ethanol and CO2. Okay, that by anaerobic respiration and process is called alcoholic fermentation. The process of fermentation in which alcohol and CO2 is the byproduct of the process, then the process is called alcoholic fermentation, which runs under an aerobic condition. Okay, so the carbon dioxide gas accumulate inside the meshwork of gluten, making the dough rise. Okay, when you do the baking, you will find that your bread which is not baked yet, okay, it become fluffy, it become rice, and after you do baking it will remain the fluffiness present and it is become more fluffy, because when heat is applied the CO2 wants to come out but it is not properly coming out and it gives you this fluffy texture. Okay, you can see here this fluffy texture which is present inside your bread. It is because of the CO2 coming out from this batter. Okay, the CO2 which is coming out from this batter is coming, giving you this texture. Okay, so when dough is baked, CO2 escape, making the bread soft and porous. The soft and porous texture of this bread, it is coming because when you cook the bread, CO2 get escape out and it giving you soft and porous texture. Okay, so yeast is also used in some delicious items like pizza, bread, donut, croissant, cinnamon buns, bagels, etc. Okay, different assortment of bakery items are made using same process of fermentation. Everywhere in bakery the same process of fermentation is used, in which the yeast is used. Okay, मुंह में पानी आ रहा होगा देख के, donut, bread, croissant, okay, all these are the bakery product which is produced by the activity of yeast, that is your anaerobic respiration, in which the byproduct is ethanol and your CO2. Okay, next, did you know, now this is some extra knowledge. Okay, ancient Egyptian bakers and brewers, kneading and baking, they use their their hands and legs. Okay, harvesting grapes and making wines. Okay, so Egyptian used to bake bread and were also known as brave wine. Okay, you normally know that by the help of fermentation you will produce wine, alcoholic drinks. Okay, but that Egyptian use baked bread. Okay, baked bread they will use on the place of grape, that's they called as brew wine. Okay, you know wine is produced by a fermentation of grapes. Okay, everyone know wine, that grape wine. Okay, in which you use grape as a fermented product, as a raw material you use grape and do fermentation of that grape. Okay, but Egyptian people take what? They use baked bread and do fermentation of that baked bread and produce brew wine. Okay, the same alcohol fermentation process is used to make many beverages around the world today. The same, so that's how old is our bever industry. Okay, Egyptian people uses that thing, and that thing is today now it is used to produce different alcoholic beverages. Either it is whiskey, brandy, rum, beer, wine, all these things are produced in our beverage industry, and it is because it is started by Egyptians in your ancient Egypt. Okay, so food preservation. Okay, so your bacteria is also used to do food preservation. Okay, to ensure food security, fermentation was used along with the other techniques to preserve food till the next harvest season. You will put that food, okay, and added some preservative, which is a result of fermentation. Okay, and it is makes save your food for next season. Okay, for making wine, the grapes were collected, crushed to extract juice, and allowed to ferment for a long time, which is followed even today. Okay, today you will produce wine. Okay, and the process is normal. You will collect grape, you will crush them, you will put them under anaerobic condition, in which it will convert into wine. The syrup of that grape convert into wine, which you drink. Okay. Even today, fermentation is an important part of culinary science as it enhances flavor, preserve food without refrigeration, and provide good health and benefits. You eat pickle. Okay, pickle, it is also a product of fermentation. In pickle, you will see that fermentation is also there. Okay, just like beer and wine, every culture has its own type of fermented beverages. Okay, that fermented beverages which you drink, it is a result of fermentation. Okay, so here is written, just like beer and wine, every culture has its own type of fermented beverages. Different cultures, you will find different fermented drinks. In South India, you will find toddy. Okay, that is again a fermented drink. In Rajasthan, you will find kechri. Okay, or kanchi, that is again a fermented drink in which we use carrot, that chili and poppy seed to do fermentation and make that kanchi. Okay, so in different areas and different cultures, you will find different fermented drink, and but the process of that is similar, is that anaerobic fermentation, alcoholic fermentation by using yeast. Okay, so this is one of that is your toddy, which is drink in South Indian basically. Okay, so number of traditional drink and food are also made by fermentation by microbes. Toddy, a traditional drink of some parts of South India, is made by fermenting sap of palm tree. Okay, this is your palm tree, the sap, okay, the resin, the sap which is coming out from the stems of this tree is being used and fermented to produce a drink, an alcoholic drink, which is called toddy. Okay, so the people climb up to the tree and cut the base of the leaf. Okay, just this is a leaf. Okay, and the base of the leaf is being cut down. Okay, so the pot are tied there to collect the sap. Okay, just like this is a leaf. Okay, this is a leaf base, and here is your pot. Okay, so the people cutted this part halfly, and the sap which is coming out from this base of the leaf has been accumulated inside this pot, which is hanging down to this leaf part. Okay, so the pods are tied there to collect the sap. Yeast is present on the palm tree, on the human skin, and even in the ear. So where from this yeast is coming? It is present in the ear. It is a microorganism. In the starting of the chapter, I told you that yeast is present, that microorganism is present all over the world. Okay, it can be not seen by naked eye, but it is roaming. In this room now, microorganisms are present, but I cannot see by my naked eyes. Okay, so microorganism is present everywhere. On my skin, microscopic organisms are present. In my mouth, microscopic organisms are present. So microbes are present everywhere. Okay, so pores are tied to the collect the sap. Yeast is present on the palm tree, on the human skin, and even in the ear. This yeast ferment the sap naturally, and the fermented drink is ready to serve fresh in few hours. Okay, within hours, within the time of 6 to 7 hours, you get a fermented beverage which is called toddy. Okay, so it is a traditional drink drinked in South India mostly. Okay, so first now we have some question time. Okay, we have one match the following. Okay, so in this we have curd. Okay, so curd is produced by lactobacillus, that lactobacillus acidophilus. We will solve this. Okay, so first we have curd. Okay, so curd is produced by which organism? Your lactobacillus lactis or lactobacillus acidophilus. Okay, now next is your bread. okay, brewer industry, produced by what organism? That is your Saccharomyces cerevisiae or your baker yeast. Okay, now take next one. Swiss cheese produced by which organism? Okay, Swiss cheese is produced by a bacterium known as Propionibacterium shermani. Okay, and the last one is your कौन सा कलर लें बताओ बताओ येलो. Okay, Roquefort cheese produced by which organism? That is your fungus. Roquefort, that is your Penicillium roqueforti. Okay, simple question. Curd produced by Lactobacillus lactis or your Lactobacillus acidophilus. Then your bread produced by Saccharomyces cerevisiae yeast. Okay, then your Swiss cheese produced by Propionibacterium shermani. Okay, a bacterium. Then your Roquefort cheese produced by Penicillium roqueforti. Okay, so this is the match the following question which you have. This is very simple. Okay, it can be asked in your MCQ part in the board's exam. Okay, so these are the products which we eat daily, which is produced in our house, which is the household product which is produced by your microbes. That is your curd, yogurt, cheese, dhokla, pickles, dosa, idli, buttermilk, smoothies. All these things are produced by the activities of microbes in our household. Okay, so this is the flow chart at the end. Okay, microbes in household product. So these are the four products. Okay, curd, cheese, dog, and toddy. Okay, so curd, lactobacillus bacteria. Lactobacillus lab produces acid that coagulate and digest milk protein. Improve nutrition quality of curd by increasing vitamin B12. Check diseases causes microbes in your stomach. Okay, automatically this lab bacteria decreases the number of bad bacteria inside your gut. That's why it is checking the disease causing bacteria inside the curd. Okay, so this is a summary for your curd. Now second, cheese. Okay, cheese ki summary. So Swiss cheese produced by Propionibacterium shermani. Then your large hole in Swiss cheese is due to the CO2 released by fermentation. Then your Roquefort cheese produced by your sheep milk. Okay, and the organism which is used is Penicillium roqueforti. Then it gives particular flavor to the cheese. The third cheese which is not given here, that is your Camembert cheese. Okay, produced by milk cow, and the organism which is used to produce that Camembert cheese is Penicillium camberti. Okay, third is your dough. Just like idli, dosa, bacteria is used in bread. Saccharomyces cerevisiae, baker yeast is used, and toddy, fermentation of sap from palm tree. Okay, so this is the summary, last summary. Okay, microbes in food and beverages. Saccharomyces cerevisiae for toddy and bread. Penicillium roqueforti for Roquefort cheese. Lactobacillus species for idli, dosa and curd. Penicillium camberti for Camembert cheese, and Propionibacterium shermani. So this is microbes in your household. The use of microbes in your household. Again, we are going with the summary. That Saccharomyces cerevisiae in toddy and bread. Penicillium roqueforti for Roquefort cheese. Lactobacillus species which produces idli, dosa and curd. Then Penicillium camberti used to produce Camembert cheese, and Propionibacterium shermani used to produce Swiss cheese. Okay, so this is the first part of this chapter. Okay, in which I tell you microbes in household. In the next part, we are going to study about microbes in industrial. Okay, what is the act, what is the use of microbes in industry. Okay, for now, thank you for watching. Have your lunch. Okay, and दिगु then दिगु नमा तपस्या and कभी झुकने का नहीं. Okay.